In this video, I'm going to recap the structure of glucose molecules, the two different isomers of it, alpha and beta, and also focus on how these molecules can be built up into polysaccharides and the difference between their structure, how it relates to their function. We know that carbohydrates are an essential part of a balanced diet. If you consider the basic word equation for respiration, glucose is your key substrate, and it involves oxidizing it so that you can release energy from the food that we eat. So carbohydrates are foods which give us a store of energy. As well as that, carbohydrates also play a structural role. So carbohydrates make up cellulose, which is essential for the cell wall components of plants. They make up chitin, which is important in fungal walls and exoskeletons for certain kinds of insects, and also make up the peptidoglycans within bacteria as well. Other functions that you will have come across, sucrose is a type of carbohydrate, which is how carbohydrates are transported when glucose is made in photosynthesis. It's converted into sucrose and transported in that format throughout the phloem of a plant cell. At GCSE, you learned that carbohydrates can be broken down into their constituent parts, i.e. basic sugar molecules, and that's done through the action of carbohydrase enzymes. Your key example you will have covered is amylase, which breaks down starch, one type of carbohydrate, into glucose molecules, another type of sugar. What we're saying as we move into A level is that glucose is not the only simple sugar. There are others, but there are certain things that all carbohydrates have in common with each other. All carbohydrates contain only carbon, hydrogen, and oxygen. And when you consider the simple sugars, the monosaccharides that make these carbohydrates up, you are always going to have the same number of carbon as oxygen atoms. Typically, carbohydrates have the formula CH2O and then a little n at the end of it. So the carbon and oxygens are the same as each other. You've normally got double the number of hydrogens, and how many of those units there are depend on which kind of carbohydrate you're talking about. Now, N, for a simple sugar, a monosaccharide, can be anywhere between 3 and 7. So you can have triose monosaccharides that only have 3 carbons, and you can have hexose monosaccharides that only have 6 carbons. Your key example of that you will have come across before is the molecule which contains C6H12O6, commonly recognized as glucose. All monosaccharides tend to form white crystalline solids, and they all will dissolve in water to form sweet-tasting solutions, so they're all quite soluble. As we move into A-level biology, we're not just considering the chemical formula of glucose as C6H12O6, we're also considering how are those atoms actually bonded together, because that makes a difference to what their properties are going to be. Now, this diagram is a visual representation of the structure of what glucose actually looks like. So what you will hopefully see when you look at this is a hexagonal shape. And at each of the 6 corners of the hexagon, we start with an oxygen atom at this corner. And then going around clockwise from that oxygen atom, I'm going to number the carbons so that I can identify which carbon I'm talking about. So I've got 1 carbon here, I've got a 2nd carbon here, a 3rd carbon here, 4, carbon 5, and carbon 6. So there you can identify where the 6 carbons from C6H12O6 actually come from. The reason I'm numbering them and why it's important to number them in this way, i.e. you start with looking at oxygen in that corner of the hexagon and then move around clockwise, is because the position of the carbons can be used to identify where bonds are actually going to form. So, for example, a 1,4 glycosidic bond is a bond between 2 monosaccharide units between carbon 1 of one atom and carbon 4 of the next. More of that in just a moment. Looking at this diagram, we can also see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12 hydrogens. And 1, 2, 3, 4, 5, 6 oxygens. A lot of biology at A level relies on you remembering key facts, and there are a lot of key facts to remember. Being able to draw this diagram of glucose and identify where all of the oxygens and hydrogens are is an important fact to remember, and you're going to keep coming back to it. Now would be a good time to pause or turn off your screen and see if you can actually draw this molecule out yourselves without looking. Then look at the screen again. What did you miss? As with all repetition and memory, the more often you do it, the easier you will remember this. One thing that will help is remembering that all of these atoms are not arranged haphazardly. They're in functional groups. And you will see that from each carbon, for carbons 1, 2, 3, and 4, each carbon, when it's not part of the hexagon itself, is bonded with one hydroxyl group, an OH group, and one hydrogen group. One hydroxyl group, one hydrogen group, hydroxyl group, hydrogen, hydroxyl group, hydrogen. So it helps to remember for these 4 carbons that what's coming off them is a H and an OH. The presence of the hydroxyl groups, i.e. the OH groups, is what makes a molecule polar, and it makes it soluble. So it's important to know exactly where those hydroxyl groups are. We can draw a simplified version of this glucose molecule when we want to just focus on the groups which are involved in bonds, and we want to ignore the rest because for the purpose of what we're showing, it's not as important. I can simplify this diagram of glucose by showing the hexagon with only oxygen in one corner, knowing that carbon molecules complete all the other corners. I'll just draw a little bond up here to show that you've got CH2OH coming off there as well. But the only other functional groups I'm actually interested in is what's coming off carbon 1 and carbon 4. So here I have OH group there with a H group coming down, and on this side from carbon 4 I have a hydroxyl group coming down and a hydrogen group coming up, which matches the way this structural representation is. Here are 2 different isomers of glucose. Now, a stereoisomer is a version of that molecule that has the same chemical formula, and the basic bonding structure is also identical, but what's different is the orientation of the bonds. Pause the video for a moment and see if you can spot the difference between the diagram on the left and the diagram on the right. Hopefully what you noticed is, if I go around and number the carbons, carbon 1, 2, 3, 4, 5, 6, on each side, when you play spot the difference you should be able to see is that on carbon 1, the hydroxyl group is at a slightly different orientation. With the first diagram on the left, the hydroxyl group on carbon 1 is going downwards, it's below the plane of orientation, and on carbon 1 on the diagram on the right, it's going up. So we have 2 different versions of glucose. Everything else is the same. The version on the left with the first hydroxyl molecule coming down is called alpha glucose, and the version on the right is called beta glucose. And the fact that that hydroxyl group is in a slightly different orientation changes the properties of these 2 molecules to the point where it affects what kind of bonds they can actually make, and therefore the overall structure and function of certain polysaccharides when you put these together. It is important to be able to draw and identify the difference between alpha and beta glucose. What might help is to think about the orientation of these hydroxyl groups. With alpha glucose, your hydroxyl groups first of all are down, then up, then down, 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 up, down. With the second glucose, the beta glucose, if you remember, beta balloons upwards. So for the first carbon, the hydroxyl group starts at the top, not at the bottom. It starts going up. So instead of down, down, up, down, when you think about where the hydroxyl groups are with beta glucose, it's up, down, up, down. Again, the more often you draw this out yourself, the more often you look at it and remind yourself, the easier you will remember this key fact. Consider this simplified version of one of those 2 glucose isomers. If I start with looking at carbon 1 and then go around and look at the hydroxyl groups, they're going down, down, up, down, along the plane of orientation as you go from carbon 1 to carbon 4. So because of that, this is an alpha glucose molecule. When a condensation reaction occurs between two glucose molecules, alpha glucose in this case, then that condensation reaction can happen between carbon 1 of one of these molecules and carbon 4 of the second molecule. So when those two come closer together, as they come closer together, a water molecule, i.e. H2O, is released and a covalent bond forms between the remaining oxygen and the adjacent molecule. Now, overall, we call this a condensation reaction, and this covalent bond that has been formed connecting these two monosaccharides is called a glycosidic bond. When you have two alpha glucose molecules forming a disaccharide, a carbohydrate with two monosaccharides, as in this case, we call this molecule maltose. When you have multiple glycosidic bonds forming between carbon 1 and carbon 4, what you find is that because of how the bond actually forms, the overall molecule starts to form a helical structure. And when that continues over an extended number of molecules, you end up with a helical structure of a polysaccharide, which makes it quite compact. We call this structure amylose. So amylose is a polysaccharide, as in it's made up of many monosaccharide units. It's made up of many alpha glucose monosaccharide units, which are joined together with 1,4 glycosidic bonds because they're forming between carbon 1 of one monosaccharide and carbon 4 of the next one. Because of their helical structure, amylose is a good way of storing a lot of energy because you can pack in lots of glucose molecules into this helical structure. As well as 1,4 glycosidic bonds, you can also get 1,6 glycosidic bonds forming between alpha glucose molecules as well. So what that means is, if I show you the number of the carbons again, you've got carbon 1, 2, 3, 4, 5, 6. And on this one, carbon 1, 2, 3, 4, 5, 6. This is a covalent bond forming between carbon 1 and carbon 6. So again, a water molecule has been released. This is a condensation reaction, but what it does is form a branching off point. So instead of just one long helical chain, you can now get a structure that has branches coming off it. Starch is typically a mixture of the amylose component, which is where you have the helical structure, and the amylopectin component, where you have branches coming off. So each one of these branches is where you've had a 1,6 alpha glycosidic bond forming. We already mentioned that having a helical structure means that amylose can store a lot of glucose molecules in a small amount of space, i.e. it's a compact molecule, but having all of these branches mean that amylopectin is good at releasing glucose molecules when they're needed, because every single one of these branches is where a glucose molecule can be removed at the same time. So the more branches it has, the more energy can be released. Now, starch is found in plants, and plants don't really have a really high respiratory requirement. Animals need a lot more glucose molecules to be released in a shorter period of time because we have a higher metabolic demand. Plants store excess glucose molecules as starch, which is a mixture of amylose and amylopectin. Animals store extra glucose as glycogen. And what you can notice is the two have certain similarities. They are both made up of alpha glucose. They both have 1,4 glycosidic bonds, which give them a helical structure. They both have 1,6 glycosidic bonds, which give them the branching structures, but glycogen has a lot more branching off points than amylopectin does. And because it has more branching off points, there are more places where glucose can be released quickly through hydrolysis reactions to ensure a fast release of glucose for respiration when it's needed, which makes sense because, again, animals have a much higher metabolic demand. They need to be able to carry out more respiration faster than plants do. The final polysaccharide we need to cover is cellulose. And cellulose is made up of not alpha glucose, but beta glucose molecules. And we can see that by looking at this diagram, because instead of down, down, up, down to represent the orientation of the hydroxyl molecules, here from carbon 1, the first hydroxyl molecule is going up, ballooning upwards for beta glucose molecules. This is important because it makes a difference to how it can form 1,4 glycosidic bonds. When two beta glucose molecules orient themselves to try and form 1,4 glycosidic bonds, you can see that between the hydroxyl group from carbon 1 and the hydroxyl group from carbon 4, there's too much of a difference because they're on different planes of alignment. So in order to actually be able to form this bond, one of these glucose molecules has to flip upside down. And when it does that, you can now see that we've got two hydroxyl molecules right close to each other, which means that, again, it's easy for H2O to be removed and to form a covalent bond between the carbon 1 of one of these atoms and carbon 4 of the other one. Then the next molecule comes and lines up, and this time, because the middle molecule had flipped, we've got our two hydroxyl groups really close to each other, and so your covalent bond can form here, and water molecule is released again in a condensation reaction. When you have lots of beta glucose molecules forming bonds in this way, where every other beta glucose molecule flips upside down in order to allow these condensation reactions to happen, it means you end up with a very straight line structure within the polysaccharide. That straight structure means that overall the cellulose molecules, which again are made up of beta glucose molecules, so they've still got 1,4 glycosidic bonds, but we call them beta 1,4 glycosidic bonds because it's between beta glucose, not alpha glucose. And because it's beta glucose and you have every alternate molecule flipping upside down, overall it has a very long straight chain structure, which is important because cellulose needs to give the cell walls structure and support. So in each of these cases, we can see how the structure relates to the function of these polysaccharides.